This isn't science fiction. There are currently hundreds, perhaps thousands of test plots of GE trees already being grown in the open around the world. My name is David Suzuki. I am, by training and profession, a geneticist. And for 25 years, I had an active career in science, once having the largest genetics lab in Canada. I'm narrating this film because I'm concerned about the unseemly haste with which my colleagues and my peer group seem to be ready to rush in and begin to apply ideas in this revolutionary area, to apply ideas that I think are far too early to expose people either in our drugs, in our food, or out in open fields. Genetic engineering is the insertion of genetic material, or DNA, into the cells of a living organism, usually using a bacterium or a virus. The artificially inserted DNA, selected for a specific trait of interest, can be extracted from an entirely unrelated plant, animal, or microorganism. The result is a plant or tree that has been altered in a way that could never occur in nature. Nor could it be achieved through traditional cross-pollination or hybridization. Genetic engineering is a new and complicated field, which often yields unexpected results. In discussions I've had with my fellow geneticists, they often say, listen, Suzuki, we're just talking about DNA. DNA is DNA. What difference does it make what organism it comes from? We pull it out of this organism, put it into another, it's just DNA. They forget a fundamental fact. We study the genetics of organisms by breeding a male and a female of one species, looking at their offspring and breeding them through what is called vertical inheritance within a species. When you take a gene from one species and transfer that DNA into a totally unrelated species, that's a completely different kind of experiment. This is now called horizontal inheritance. We've never done that before, and it is absolutely bad science to say that we can look at vertical inheritance and use the same ideas to explain what goes on in horizontal experiments. It's just lousy science. Biotechnologists think genes are genes. It doesn't matter where you stick them, and they'll just function the way they normally do. Any geneticist who thinks about that should know better. Genes don't function alone. They function within the context of the entire genome. Nature acts on the entire genome because after fertilization, there are whole sets of genes turned on and off in the proper sequence so that you get the development of a complete organism. So that whole orchestration is an integrated genome that acts as a complete entity. To take a gene out of a fish and stick it into a plant means the fish gene suddenly wakes up and goes, where the hell am I? Who are all these other genes around me? Because you've altered the context within which that gene is found. It would be like taking Bono out of U2 and putting him into the New York Philharmonic Orchestra and say, okay, folks, play music. Well, you get noise of some sort, but nobody can anticipate what the sum total of that activity will be. It's just a mistake to think that genes act as if their traits are expressed regardless of where they exist.
There's debate over the direct threat of the inserted genes in GMO crops, as they may not be detectable in the human body, but the real danger may come from pesticides associated to genetically modified foods. Genetically engineered seed biotechnology typically has not been used to increase crop yield, nutrition, drought tolerance, but instead for profitable pesticide-resistant products. 80% of the GMO crops are bioengineered only for pesticide resistance. Not surprising, given that the top 5 biotech companies are chemical companies that manufacture pesticides. This allows farmers to spray herbicides directly onto the crops, raising a theoretical possibility that the level of residues of the herbicide on food we buy at the supermarket may have increased. Or at least it was theoretical until now. Monsanto's Roundup Ready soybeans are the #1 GM crop genetically engineered to be resistant to the herbicide Roundup, also sold by Monsanto, allowing farmers to spray fields with the Roundup herbicide glyphosate, which then kills the weeds while leaving the soy standing. Monsanto maintains that Roundup-Ready soybeans are compositionally equivalent to that of conventional soy, a concept that is used to argue that GMO foods are therefore safe as non-GMO. But Monsanto didn't report the level of pesticide residues. In fact, some of the comparison tests were done on Roundup-Ready soybeans that hadn't been sprayed at all, which is the whole point of having Roundup-Ready plants, so you can spray them with Roundup. In contrast to real-life samples from the market, transgenic crops intended for scientific studies are often produced without the application of herbicides, or at doses lower than typically used by farmers. It wasn't until this study was published in 2014 when the full composition of ready-to-market soybeans was analyzed. Here's how much glyphosate was found in the GMO beans, along with a glyphosate breakdown product called AMPA. Here's how much was found in organic soy. None. What about conventional non-GMO soy, where glyphosate is sprayed on the soil to kill weeds between crop cycles? None. So GMO soybeans are really not equivalent. They appear to have substantially more pesticide residues. The debate then shifts from the safety of Roundup-Ready soybeans to the safety of Roundup itself. Are those glyphosate residues on GMO soy something to be concerned about? A question.
Attack on scientists is very well structured by the biotech industry. It's systematic, it's worldwide, it's very coordinated.
Our discovery that we were finding transgenic corn maybe 1,000 miles from the nearest legal transgenic corn field was a huge problem for them, because it really showed very simply and with real evidence that they really did not have control.
The list of frauds and abuses continues to mount, from concerns over the introduction of Bt brinjal in India, to the mysterious appearance of GMO wheat in supposedly non-GMO crops in the western United States, to the appearance of unapproved GM corn in shipments of supposedly non-GMO corn to China just this week. These types of problems have led to the outright refusal of a growing number of countries to accept genetically modified crops. But in a world where the issue of containment of GMO crops is increasingly called into question, and where even the ability to label GMO foods may be stripped from the people altogether thanks to overarching closed-door agreements like the Trans-Pacific Partnership currently under negotiation between the US and 11 of its Pacific Rim counterparts, these issues are becoming increasingly pressing. And if critical science, like that conducted by Seralini's team, can be so easily sidelined, dismissed, and retracted, what does that say about the system that is currently in place to ensure the validity and accuracy of food toxicity studies? As a team of independent scientists and researchers from across the world wrote in an open letter last year during the eruption of the Seralini affair, when those with a vested interest attempt to sow unreasonable doubt about inconvenient results, or when governments exploit political opportunities by picking and choosing from scientific evidence, they jeopardize public confidence in scientific methods and institutions, and also put their own citizenry at risk. Safety testing, science-based regulation, and the scientific process itself depend crucially on a widespread trust in a body of scientists devoted to the public interest and professional integrity. If instead the starting point of a scientific product assessment is an approval process rigged in favor of the applicant, backed up by systematic suppression of independent scientists working in the public interest, then there can never be an honest, rational, or scientific debate.
There's been a bit of an uproar this past week due to Obama's signing of a recent bill that has become affectionately labeled the Monsanto Protection Act, or H.R. 933. So much so that it's led Senator Barbara Mikulski to publicly apologize for letting the provisions slip through. However, this is really only a small part of a much bigger pattern of corruption and collusion between Monsanto and the U.S. government. Senator Roy Blunt, a Republican from Missouri, who crafted the wording for H.R. 933, has been accused by Senator John Tester of having collaborated directly with Monsanto in the creation of the provision. This shouldn't be surprising considering that Monsanto is one of Blunt's top campaign contributors. Blunt also accepts contributions from the Crawford Group, a registered lobbyist for Monsanto. Now, the controversy in this bill is tied to Section 735, which is of course written in such a jumble of incomprehensible legalese that it's highly difficult to interpret. It's designed to make the eyes glaze over without registering that anything of significance has been stated. This is facilitated using a common legislative technique of referring to sections of a separate law, so that you have to go read the unintelligible fine print in that second document in order to understand what's actually being said. That second law in this case is the Plant Protection Act. In the description of this video you'll find a link to both documents. Now, what Section 735 of H.R. 933 states is that if a plant is determined to be a biological pest and is therefore brought under regulation and restrictions which prevent it from being planted or transported within the U.S., the Secretary of Agriculture shall immediately grant a temporary permit or deregulation to any farmer, farm operator, or producer that requests it, regardless of what any other laws or courts may order. Now, the word genetically modified organism isn't mentioned here, and that's part of the skillful way that this was designed to slip through. Only someone who knows that by default genetically modified organisms are considered biological pests until they've been approved by the Department of Agriculture, and that they are reclassified as pests if shown unsafe, would understand the implications here. To distill this down to one sentence, if a farmer wants to grow a genetically modified plant that's been outlawed because it's been found to be harmful to the environment or human health, all they need to do is contact the Secretary of Agriculture, and they will be immediately issued a permit to cultivate these plants. Essentially, it makes it impossible to stop a genetically modified plant from being used in the United States, regardless of any laws or rulings that are made to outlaw them. Now, of course, one might be inclined to let Obama off the hook for signing this law. After all, it is written in legalese, and one could presume that he just made an innocent error. But this would require overlooking the extensive evidence of the Obama administration's incestuous relationship with Monsanto, among which include his appointment of Michael R. Taylor, former Vice President of Public Policy for Monsanto, as his FDA Deputy Commissioner for Foods and Veterinary Medicine, his appointment of Roger Beachy, former Director of the Monsanto Danforth Center, into a position at the USDA as the Director of the National Institute of Food and Agriculture, and his choice of Islam Siddiqui, former Monsanto lobbyist, as his Agriculture Trade Representative. Now, anyone who thinks that the revolving door between the government and Monsanto is something new should look into Monsanto's connections and dealings with both of the Bush administrations and the Clinton administration. This has been going on for a long time.
In the U.S., farmers, knowing their crops can tolerate Roundup, have tripled the use of the herbicide, according to the U.S.-based Pesticides Action Network.
If you take plants and make them resistant to an herbicide, what does that mean? It means, hey, no problem, you can put herbicide all over the place and it won't kill the plant. And so, of course, it's associated with greater herbicide use.
Northwestern Iowa, that's where I'm from. And he told me that he was growing genetically modified corn side by side with non-genetically modified corn. And then decided to do an experiment. He took a trough and put the GM corn on one side and the non-GM on another, and then all of his cows into the pen. And they all huddled around the non-GM side and wouldn't eat the genetically modified side. So he started telling other farmers about it, and they started doing their experiment, letting cows into a pen 2 or 3 at a time. And this time they had completely separate troughs, GM and non-GM. They'd go up to the first one and they'd taste it or smell it, but then they'd stop and they'd walk around it and go to the non-GM trough, eat all the corn there, and then go back to the first trough and taste it and then walk away. So animals, when given a choice, often choose non-genetically modified foods. You may not know it, but you eat genetically modified ingredients in almost every meal. We don't have the sixth sense of the animals to avoid GMOs naturally. We don't have the labeling of GMOs, which is enjoyed by Europeans, Australians, Japanese. Most of the rest of the world label GMOs, but we don't. Why is it put on the market without labeling? Because the policy at the FDA claimed that they were unaware of any information showing that GMOs were significantly different. But years later, thousands of secret memos were made public from the FDA's files, and it showed that that policy was a lie. But the person in charge of policy was Monsanto's former attorney, later Monsanto's vice president. Here's the great news. We have the power. In Europe, they kicked out GMOs. When a small percentage of the shoppers said, we don't want to eat GMOs, within a single week, virtually every major food company said, we're not going to use GM ingredients anymore in Europe. But those same companies use GMOs in the United States, where most people haven't raised a fuss. So here's what we're going to do. We're going to raise a fuss.
Biology. From matter arising. Complexity emerging. Megamolecular concatenation. Protein synthesis. replicated self-similarity, a bioholographic self-image, propagating through space-time, the bio-urge, an impulse toward free and undeterred action, becoming sensation, compelled by tropism, soaking up light and heat, energetic attraction of chemistry, replicating its way on the long, long journey toward biology. From gravity emerging, and the attraction of like for like, the cataclysmic explosions of energetic connectivity, forced together against the repulsive tide of insensate crystallographic repetitiveness and fractal chaos. Inchoate densities of molten molecular matter. Complex proteins form, and then, 4 billions of years, 4,000 million years ago, a momentary coalescence, a circumstance of coincidence, the threshold of life in that vast unknown space, singular organisms, the earliest cells dividing.
Most of our current ideas, especially in a revolutionary area like biotechnology, are going to turn out to be wrong. That's not a denigration or an insult to science. That's the way a science progresses. You make a set of observations, you set up a hypothesis to make sense of them, you carry out an experiment, you get the results, then you say, how well did we do? Chances are you say, oh man, was I wrong? You toss that hypothesis out and set up another one. That's how science progresses. So I want to repeat, in any revolutionary area, and biotechnology is a revolutionary area, most of our current ideas are wrong. Then I ask you, what the hell is the rush to apply these ideas that we're getting? We're still at the bare beginning of understanding of what we're doing. The rush to apply these ideas is absolutely dangerous because we don't have a clue what the long-term impact of our manipulations is going to be.